Microbial Metabolism, Part 2. This is an overview that shows the three major stages of cellular respiration: glycolysis, the transition step, which is not really considered a stage, but it's very important, the Krebs cycle, and electron transport oxidative phosphorylation stage. You should be very familiar with how this system works and the molecules produced at each stage. I'd like to point out that there are very few ATP molecules that are formed during the first two stages, and the majority of the ATP molecules are produced during the final stage of electron transport and oxidative phosphorylation. You should refer back to this slide while studying. It's important to note that there are many reactions that take place during these three main stages, and each reaction uses a different enzyme. These reactions are not pictured here because you don't need to memorize each enzyme or each reaction. The point here is to understand what is going on in the cell and why it's happening. The first step of glycolysis is the most important step of all, because as glucose enters the cell, phosphate groups from two ATP molecules are transferred to the glucose. Recall from biomolecules that phosphate groups has a negative charge. So when added to the glucose molecule, it gives the glucose a negative charge, so that if the glucose tries to leave out of the cell, it is repelled by the negative charge on the phospholipids of the cell membrane and remains inside of the cell. So this is why this first stage or this first reaction of glycolysis is the most important, when the glucose is gaining those phosphate molecules. Because without the phosphate groups, the glucose molecule could just leave back out of the cell and cellular respiration would not occur. So in the first part of glycolysis, two ATP molecules are used, which is why it's called the energy investment stage. At the end of this initial phase of glycolysis, the 6-carbon glucose molecule is split into two 3-carbon molecules that enter the second phase of glycolysis. By the end of this phase, 2 molecules of pyruvate and 2 molecules of NADH are produced. Also, 4 molecules of ATP are made, but because we used 2 molecules of ATP in the beginning of glycolysis, there is only a net gain of 2 ATP molecules. As a reminder, ATP, or adenosine triphosphate, is the energy source for the cell. Its structure resembles that of the adenine nucleotide, but instead of having one phosphate group, it has 3. When the third phosphate group is removed, energy is released that can be used in the cell to do work. The removal of the third phosphate results in adenosine diphosphate, a molecule with 2 phosphates. That second phosphate can also be removed to release energy, but less energy is released from ADP than is released from ATP. ATP utilization and replenishment is a constant cycle in living cells. ATP is constantly used for energy to do work in the cell, and then the ADP that results can gain a phosphate to replenish the ATP molecule. ATP is produced by 2 mechanisms during cellular respiration: through substrate level phosphorylation in glycolysis and the Krebs cycle, and by oxidative phosphorylation in the electron transport chain, which we will discuss later. This figure shows how substrate level phosphorylation works. A substrate that contains a phosphate group transfers its phosphate to ADP to make ATP. It's that simple. At the end of glycolysis, two molecules of pyruvate are formed. Pyruvate, also known as pyruvic acid, is an intermediate product that can be used in a number of different metabolic pathways. It can be used to synthesize amino acids, carbohydrates, and lipids. It can also be used by some organisms in fermentation reactions. Right now, we'll see how it's used as we continue through the next two stages of cellular respiration. Pyruvate still has the original 6 atoms of carbon that came from the original glucose molecule. The molecule has only split in half. During the intermediate step and Krebs cycle is when the molecule starts to be degraded and will lose all 6 of the carbons in a stepwise fashion. Both pyruvate molecules enter the intermediate step to be converted to the molecule acetyl-CoA. During this step, 2 molecules of carbon dioxide are also released, one for each molecule of pyruvate, and 2 molecules of NADH are formed. We'll learn more about what those NADH molecules are used for a little bit later. At this stage, 4 carbon atoms are remaining from that original glucose molecule, 2 in each molecule of acetyl-CoA. Both molecules of acetyl-CoA enter the Krebs cycle, and each molecule is combined with the molecule of oxaloacetic acid, a 4-carbon molecule. Together, the acetyl-CoA and the oxaloacetic acid combine to make citric acid. The citric acid begins the Krebs cycle, and the next 2 reactions result in a release of carbon dioxide molecule. And after reaction number 3, all of the original carbon atoms have been removed in the form of carbon dioxide. The remaining 4-carbon molecule continues through the Krebs cycle in order to regenerate oxaloacetic acid, so the Krebs cycle can continue with the next set of acetyl-CoA molecules that enters the cycle. The products of the Krebs cycle are 6 molecules of NADH, 2 molecules of FADH2, which is another hydrogen-carrying molecule like NADH. 2 molecules of ATP are also formed, and 4 molecules of carbon dioxide. During the electron transport chain, the hydrogen atoms that are bound to the NADH and FADH2 are used to produce ATP by the oxidative phosphorylation method that is powered by chemiosmosis. The electron transport chain is made up of a number of proteins that are embedded in the cell membrane of prokaryotes or in the mitochondrial membrane of eukaryotes. The hydrogen atoms that are carried by the NADH and FADH2 are donated to the electron transport chain to generate energy needed to make the ATP. This slide is a summary of events that occurred during electron transport. But let's see how it works. In this figure, you see the cell membrane of the prokaryotic cell with proteins of the electron transport chain embedded within them. The large blue structure on the right side of the cell membrane is the enzyme responsible for production of ATP. In bacteria, NADH and FADH2 donate their electrons to the electron transport chain. On the left side of this electron transport chain, the hydrogen atom are released from NADH and FADH, and the hydrogen splits into the hydrogen proton and an electron. The electrons are transported through the membrane from one protein to the next in the electron transport chain. Each transfer of the electron is an oxidation-reduction reaction. When one protein grabs a hold of that electron, it's being reduced. When it releases it to the next protein in the cell membrane, it is being oxidized and the second protein is being reduced. And these oxidation reduction events occur as the electron moves from protein to protein, releasing energy as it goes to power other reactions that are occurring in cellular respiration. This continues, this oxidation reduction of these proteins continues until that electron reaches the last protein in the chain. The electron then re-enters the cytoplasm at that point and recombines with a proton to form a complete hydrogen atom again. Two hydrogen atoms then bond to oxygen and form water. So oxygen is considered the final electron acceptor because those hydrogens will bond, those hydrogens that are carrying the electrons bond to the oxygen, and the oxygen itself is accepting those hydrogens or electrons as we like to think of them. So this is why oxygen is very, very important in aerobic cellular respiration. It gives the electrons somewhere to go at the end of the electron transport chain because they can't remain in the membrane. If oxygen were not there, this entire process would stop because the electrons would be stuck in the membrane in those proteins and would have nowhere to go. So what about the protons? What happens to the protons? When the NADH and the FADH2 donate those hydrogens to the electron transport chain, remember those hydrogens are split into protons and electrons. We've discussed what happens to the electrons, but now let's see what happens to the protons. When the hydrogen was donated, we can see here that it was split into an electron and into a proton. While the electrons are being transported through the membrane, the hydrogen protons are being transported across the membrane, and they accumulate in the periplasmic space, that space just outside of the cell membrane. Eventually, a concentration gradient is formed where it's more positive in the periplasm than it is inside the cell in the cytoplasm. And hydrogen protons begin to re-enter the cytoplasm through that large blue structure called the ATP synthase protein. The movement of protons through the ATP synthase supplies the power to form over 30 molecules of ATP. This mechanism of forming ATP by the oxidation of NADH or FADH2 is called oxidative phosphorylation. So this is the second method in which ATP is produced in the cell. The first method, remember, it is termed substrate level phosphorylation. And this is oxidative phosphorylation because at the beginning of the electron transport chain, NADH and FADH lose those hydrogens and donate them to the electron transport chain. They are being oxidized because by losing hydrogens, they're also losing electrons. So that is the oxidative part of it. The phosphorylation part is when the hydrogens re-enter the cytoplasm through the ATP synthase, and this gives that enzyme the power it needs, the energy it needs to take ADP and free phosphates, make that connection, make that bond, and reform the ATP molecule. So this is what happens in the electron transport chain to produce those 30-something molecules of ATP. Aerobic cellular respiration is what happens when oxygen is available. Remember, we use oxygen as that terminal electron acceptor when those electrons are coming out of the electron transport chain. They recombine with the hydrogen proton, and then they reduce oxygen to form water. The first two stages of glycolysis and the Krebs cycle don't really require oxygen, but if oxygen isn't supplied at the end of the electron transport chain, cellular respiration will stop because cells need to release those hydrogens to the electron transport chain, and the NAD and the FAD needs to go back to the glycolysis stage and to the Krebs cycle so that it can capture those hydrogens that are continuously coming off of the glucose molecule as it's being oxidized. So, again, the first two stages, glycolysis and the Krebs cycle, do not really require oxygen, but they do indirectly, because if oxygen isn't supplied during the electron transport chain, cellular respiration will stop and the cell will die unless it is able to survive through fermentation or anaerobic respiration. In fermentation, the glucose goes through glycolysis and pyruvate is formed, just like normal. Then, depending on the organism and its capabilities, pyruvate can be converted to a number of different products. You don't have to commit this slide to memory, but we will talk more about lactic acid fermentation and ethanol fermentation. During glycolysis, only two molecules of ATP are formed. The purpose of fermentation is to regenerate NAD+. Remember that. The purpose of fermentation is to regenerate NAD+, so that glycolysis can continue and the organism can stay alive by continuing to make that two net molecules of ATP. Remember, 4 total ATP are made during glycolysis, but 2 ATP are used in the beginning of glycolysis, so we only get a net amount of 2 ATP molecules during glycolysis. During lactic acid fermentation, the pyruvate is reduced to lactic acid. The pyruvate gains a hydrogen atom and therefore an electron from the NADH, and then NAD+ is regenerated. The NAD can then return to glycolysis so that more ATP can be made. This cycle of glycolysis and reduction of pyruvate continues until the organism just runs out of glucose. Live active cultures in bacteria that break down the lactose in milk into galactose and glucose. These organisms then use the glucose to form lactic acid, which gives yogurt its sour taste. Alcohol fermentation is performed by yeast. The pyruvate is first converted to acetaldehyde. During that reaction, carbon dioxide is released. The acetaldehyde is then reduced to ethanol in the same manner as in lactic acid fermentation. When yeast is used to make the dough for bread, it rises because of the carbon dioxide that is released. You don't get drunk when eating bread because the ethanol is burned off during the baking process. So in alcohol fermentation, there are two steps of conversion of pyruvate to the ethanol. First, that conversion of pyruvate to acetaldehyde, and then that second conversion or reduction of acetaldehyde by the addition of a hydrogen to ethanol. And then this reduction of acetaldehyde then regenerates that NAD so that it can again be used in glycolysis to continue to make those two ATP molecules and give that cell enough energy to survive. Fermentation is a way for some organisms to survive without oxygen, and still there are others that can perform anaerobic respiration. With anaerobic respiration, the organism still goes through glycolysis, the Krebs cycle, and the electron transport chain, but instead of oxygen being that electron acceptor at the end of the electron transport chain, a number of different electron acceptors can be used, and listed here are just a few. Organisms that can do anaerobic respiration make more ATP than those that do fermentation because they still go through all three stages, but they don't make as much ATP as they would if using oxygen. So if oxygen is available, some organisms are using oxygen to undergo aerobic cellular respiration. Some organisms are able to do anaerobic respiration using electron acceptors other than oxygen. And still some organisms are able to undergo fermentation and take that pyruvate and reduce it to other final products so that NADH can be reformed and glycolysis can continue. You should be able to explain what would happen if something goes wrong. For example, if cells are growing in oxygen and then they run out of oxygen. If they're still able to do fermentation or anaerobic respiration, what would happen? So understand these three processes. Understand that aerobic respiration, oxygen is required. Anaerobic respiration goes through all the phases or the stages of aerobic respiration. It goes through glycolysis, it goes through the Krebs cycle, and the cell goes through the electron transport chain. Everything is pretty much the same except for at the end of that electron transport chain, an alternate or a different electron acceptor is used other than oxygen. Also understand that all cells are not able to undergo anaerobic respiration when there is no oxygen. Some cells will just die. Some cells can't use other electron acceptors, and some cells cannot do fermentation. But know the difference between those three processes. Cellular respiration is just the tip of the iceberg. This is a slide showing a bunch of different metabolic pathways. Okay, these can all occur in whatever cell this is representing. In red is glycolysis at the top in the rectangle. Those are the steps of glycolysis. And the Krebs cycle is circled in red here. So keep in mind that intermediates are being formed during these metabolic pathways. Just like I showed in that one slide with the enzymatic pathways with those two intermediates that are formed. So keep in mind that any intermediates that are formed during the three main stages or these two main stages can be rerouted to other pathways to make other products the cell needs to survive. and intermediates from other pathways can be rerouted into glycolysis or into the Krebs cycle. So this slide should give you a much better appreciation of what our cells and the cells of bacteria can do. Our last slide is to provide you with an overview of all three processes: aerobic respiration, anaerobic respiration, and fermentation. This is the end of Microbial Metabolism Part 2. Thank you for listening.